Okay, so we had left off talking about simple stains, which we said the purpose of a simple stain was to allow us to kind of view the basic shape of the cell and any structures that that would be visible by simply using that type of stain. Now, unlike simple stains, differential stains react differently with different kinds of bacteria, and that can help us to differentiate or distinguish them. So if you remember, we talked about differential media, right? So differential media allowed us to differentiate between different microorganisms within the media based upon color changes that we saw or production of gas, right? So it allows us to distinguish between different bacteria. So it's the same thing with a differential stain, is it allows us to differentiate between two different types of bacteria that may be on the same slide. So one of the most frequently used differential stain is that of the Gram stain, and another one is what we call an acid-fast stain. So these types of staining procedures are typically more complex, more involved, and have more chemical reagents that are used as compared to a simple stain, which we said was just basically one dye. So in differential stains, we have the primary dye, which is the one that's used initially, and then a lot of times that primary dye is washed off, and then a second dye is applied, which is called the counter stain. And this allows us to distinguish between the two different bacteria, because one will take up the primary dye and the other one typically will take up the counter stain. So as I mentioned, the Gram stain is a differential stain. So the Gram stain was developed by Hans Christian Gram, and it classifies bacteria into two large groups, what we call gram-positive bacteria and gram-negative bacteria. So in this procedure, a specimen is covered with a basic purple dye. Usually this is crystal violet. And the purple stain basically imparts its color to all the cells, and this is our primary stain. So the crystal violet is our primary stain. It's going to stain all of the cells. And after a short period of time, the purple dye is washed off, and then iodine is applied, and iodine is known as the mordant. And a mordant is a chemical that's added to the solution to help to intensify the stain. Then the iodine is washed off, and at that point we notice that both gram-positive and gram-negative bacteria typically appear purple. The slide is then washed with alcohol, or sometimes it's an alcohol acetone solution. And this solution is known as the decolorizing agent. So it removes the purple from the cells of some of the species, and it does not remove the purple from others. And this has to do with the composition of the cell wall of some of the bacteria, which we'll kind of get into later, how the gram-positive and gram-negative bacteria differ in the composition of their cell wall. Once the decolorizing agent has been applied, then we use the counter stain. In this case, it's safranin, which is a red dye, and that is applied to it. And then it's rinsed again, you blot your slide dry, and then you examine it. So the purple dye, our primary stain of crystal violet, and the iodine combine in the bacteria and cause it to be colored purple. So bacteria that retain this color are classified as gram-positive. The bacteria that lose the purple color after you decolorize it and use the counter stain of the safranin are called gram-negative, and they are kind of a pink color from that safranin. So in this image here, you can see both purple and pink bacteria. You can also tell that there's a difference in the shape of them as well. But we do have the purplish gram-positive and the more pinky kind of reddish gram-negative are visible. Another type of differential stain is an acid-fast stain. This one is a stain which binds strongly only to bacteria that have kind of a very waxy cell wall. So microbiologists typically use this stain to identify all bacteria within the genus Mycobacterium, which includes two important pathogens. You have Mycobacterium tuberculosis, which is the causative agent of tuberculosis, and then you also have Mycobacterium leprae, which is the causative agent of leprosy. So in acid-fast staining, you use a red dye called carbofusion, and you apply it to your specimen and you heat it for several minutes. And so the heating kind of enhances the penetration and the retention of the dye. Then the slide is cooled, it's washed with water, and then it's treated with an acid alcohol to decolorize it, which removes the red stain from some of the bacteria that are not acid fast. And then you use a counter stain of methylene blue. So in the image you can see kind of blue and kind of pink or reddish colors as well. So the acid-fast microorganisms retain the pink color from the carbofusion, so they'll be kind of pinkish or reddish, and the non-acid-fast bacteria are going to be blue in color. So once again, it's a type of differential stain. It helps us to differentiate between the bacteria that are on our slide. Then we have other staining procedures that we kind of classify as special stains. So it allows us to color or isolate specific parts of microorganisms, such as endospores or flagella. So the first one that we'll talk about is capsular staining. So being able to view the capsule can tell us about the virulence of the microorganism that we're looking at. So many microorganisms have kind of a gelatinous covering that's called a capsule, and the capsule can help to protect them from phagocytosis or being ingested by the host immune system. So therefore they tend to hang around much longer. So it helps us to determine the organism's virulence. In other words, the degree to which the pathogen can cause disease. So capsular staining is more difficult than other staining procedures because the capsule tends to be water soluble, so it's easy to dislodge, it's easy to kind of remove because of vigorous washing that happens during the staining procedure. So typically you're using kind of India ink or nigrosin to do a negative staining procedure where you're going to stain the background so that you can see the capsule, and then potentially using a simple stain to stain the cell itself. And then the capsule will appear kind of like as a halo, and that's kind of what you're seeing in this first image here, is that halo kind of around the cell. That is the capsule. So the background has a negative stain, and then that halo that you see is the capsule, which is a pretty thick layer that you're seeing. Then you have flagella that can be stained. So flagella are structures that are used for locomotion, so it allows the bacterial cells to move around. And these can be very hard to see with a light microscope without any type of staining at all. So flagellar staining is a very delicate staining procedure. It also can be very tedious. It uses a mordant and the stain carbofusion, which helps to build up the diameter of the flagellas to make it more visible under the light microscope. So it helps to create it, make it thicker so it's easier to see. And that's important as a diagnostic aid because microbiologists use the number and the arrangement of the flagella to help them identify the bacteria. Then we have endospores. An endospore is a dormant structure that's formed within the cell, which helps to protect the bacteria from adverse environmental conditions. So not all bacteria form endospores, but it is possible to stain them so that they can be visible under the microscope. But it's difficult to do because these are kind of resistant stages. So the most commonly used endospore stain is a Schaefer-Fulton stain. You use malachite green as the primary stain, and you heat up your slide without letting your specimen dry out. And the endospore takes up the malachite green, and then you wash it to remove any of the malachite that hasn't bonded, and then you use a counter stain of safranin, which basically stains the other structures of the cell. So if the stain has gone properly, or if it's been prepared properly, the slide, the endospores should appear green with red or pinkish cells that are around them. And I don't know how well you can see that in this image here, but at the end of the cell at one end, you should see kind of a little green oval, and that is the endospore that is within the cell, which should be kind of pinky around it, or pinkish color around it. So green endospore, and then the remainder of the cell is pink. So let's see if you can identify the staining technique that would be used for each one of these, just to kind of wrap things up and see what you've retained. So are these simple, differential, or special staining techniques? So if you were to do a capsule stain, is that a simple, differential, or special? All right, we just talked about that, and that would be a special stain. Acid-fast stain, simple, differential, or special? Acid-fast is differential. It allows you to distinguish between acid-fast and non-acid-fast, or those with those that have a very waxy material in their cell wall versus those that do not. Crystal violet would be considered to be a simple stain, right? A gram stain is a differential stain. So you'll see the difference between gram-positive and gram-negative cells. And a flagellar stain is a special stain that is used to make the flagella a lot thicker so they're easier to view under the microscope.